Welcome back to the Classics of Immunology Journal Club. We are now in the 70s, and we're going to review a paper today that is near and dear to my heart because it all has to do with long-term growth of T cells. And it's by Doris Morgan, Frank Ruscetti, and Robert Gallo, and it was published in Science in 1976. And it's entitled The Selected In Vitro Growth of T Lymphocytes from Normal Human Bone Marrow. Just to give you a little background on these people, Doris Morgan was a postdoc hired by Gallo. She did her PhD work at MD Anderson down in Texas in Houston. And before that, she was a tech for several years. So she was a little bit more mature. If you know any grad students and postdocs, she was more mature than most of them. And she was also very well skilled in cell culture, tissue culture. And she had spent her PhD time doing hematopoietic cell cultures. And that's why Gallo hired her. And he hired her on as a postdoc, one of his postdocs, but funded by his contract. He had a contract through the NCI, through a company called Lytton Bionetics, which was a former defense contractor. They did all kinds of contracting with the government because this was just after 1971 too, when President Nixon declared a war on cancer, and so this was supposed to start. You know, he was trying to reproduce the moonshot that President Kennedy had done a few years earlier. And this was going to be the thing that would give him longevity in the history of the United States of America. The thing was that the Congress didn't go along with his war on cancer, and they didn't really appropriate more grant money to do all this, but they did appropriate money that could be spent through the contracting mechanism in much the same way you'd contract to build a battleship. And so Robert Gallo, who was the head of the lab, was a lab chief at this time, and he's only 3 or 4 years older than me, which meant that he was just a whippersnapper back in the 70s. But he was already a lab chief, and he, in contrast to Doris, he went through medical school and then through his clinical training, and then he went directly to the NCI. He didn't pass go and collect $200. He went directly into cancer work, and he was interested in leukemia, in acute myelocytic leukemia, or AML. What had happened in his lab is that just a couple of years earlier, they were trying to grow leukemia cells, and they were getting bone marrow samples sent up from MD Anderson that they would then put into culture to see if they could get any leukemia cells to grow. And they were adding all kinds of sources of factors, growth factors, that they were hoping that they could get the leukemia cells to grow. And they were successful. And they got a cell line growing from one of the bone marrow samples from an AML patient that was called HL23. That was the name of the culture. And in order for these cells to grow, they needed a special growth factor, a leukemic cell growth factor that they were getting from a whole human embryo culture. And so that the cells were dependent upon this growth factor. And so Doris Morgan was hired on to find out what the growth factor was and to use the growth factor to grow up all kinds of leukemia cells so that other people in the Gallo lab could look for RNA tumor viruses, retroviruses, which is what he was after. Now, the other person in this series was Frank Ruscetti, who was also a postdoc with Lytton Bionetics in the Gallo camp. And he had done his PhD work at the University of Pittsburgh with one of the major hematology laboratories. And he had cultured human bone marrow and hematopoietic cell cultures for his PhD. So these people were not immunologists, rather they were hematologists. And Gallo was particularly interested in AML and trying to find human retroviruses. In the introduction to this paper, and this is another one of those very short papers. It's only 2 pages in Science. And I should also preface my remarks is that at that time, back in the 70s, Science was not a hotbed for biological papers from the disciplines of biology. Rather, it was much more mathematics and physics oriented, chemistry and that sort of thing. And although Gallo had an inside route to get to Science magazine, and he published a lot there subsequently. as we'll find out. So in the introduction of this paper, they point out that long-term lymphoblastoid cell lines had been established from normal human bone marrow from a lot of different normal people. The only thing is that those cell lines were all derived from B cells. They were all B lymphoblastoid cell lines. They were all Epstein-Barr virus positive, which is a large DNA virus. And basically the transformation of these B cells was dependent upon the infection by Epstein-Barr virus. And they go on to say in their introduction that there are very few long-term T cell lines, and if they were T cell origin, they were all cancer cells. Now, leukemic cell lines from patients that had T cell leukemias or T cell lymphomas. They did reference two papers, one from the mouse, from an old friend of mine, Rob McDonald from the Lausanne, who had shown with repetitive mixed lymphocyte cultures, so that you would culture together two disparate individuals, incompatible individuals, irradiate or treat one of the individuals with mitomycin C to prevent its replication, and the other person's lymphocytes would then proliferate in response to it. That's an MLC. The dogma in immunology at that point in time was that the antigen was really the critical ingredient that stimulated T cells to proliferate. And therefore, what they did in order to get cells to grow longer than just a few days was they would start the mixed lymphocyte culture, and then they'd wait a week or so, and they'd harvest the cells that were still alive and still growing, and then re-stimulate them with the original stimulator cells. And McDonald and Serratini and Brunner and those people showed that you could keep cells in culture and proliferating transiently for a week or so for months. I think they said 4 months. There was another paper out at the same time, or similarly, that was 74. This is in 75. There was a paper out from the Karolinska Institute, a fellow by the name of Svedmeyer, who did the same thing except with human lymphocytes. And he was able to show with repetitive MLCs, a repetitive antigen stimulation, that you could get cells to grow long term. And in this paper, what they say in the intro is that they didn't do that. What they did was that they put normal human bone marrow cells, and bone marrow is important in that instance, and we'll get into that, into a lymphocyte conditioned medium. The conditioned medium meaning that they would take normal lymphocytes, usually peripheral blood lymphocytes, stimulate those cells in culture with phytohemagglutinin, PHA, waited a few days, harvest the supernatant from those cultures, and that's what they use to grow T cells long term. And that's what they describe in this paper. The materials and methods were pretty simple, only two different things that you had to know about. How they made their lymphocyte conditioned medium. And by the way, they didn't talk about T cell growth factor that ultimately became interleukin-2 or anything like that. They just called this LY-CM for lymphocyte conditioned medium. So they would take, in order to make this lymphocyte conditioned medium, they would take peripheral blood mononuclear cells from people in the lab, harvest those cells from the blood, get rid of the red cells and get rid of the granulocytes, and seed those cells then at 1 million cells per ml in a 1% PHA that they bought at the store, and 1% normal human serum. They'd culture those cells then for 3 days. They'd harvest the cells, and they'd harvest the cultures, spin the cells out, take the supernatant from those cultures, and concentrate it in an Amicon filtration device about 5-fold. And then they would dialyze that culture medium against normal saline and store it at 4 degrees. They didn't freeze it. That was their lymphocyte conditioned medium. So in order to then get cells to grow in that, they would take bone marrow samples. Now, the reason they did bone marrow rather than peripheral blood cells, which is an interesting kind of thing, is that they'd already done this with the reason that they started this in the first place was that they were putting bone marrow samples from leukemic patients into conditioned medium from all kinds of sources and so forth and so on, because they were looking for a leukemic cell-specific growth factor. That's what they wanted to find. And they got cells to grow from these leukemia patients, and they didn't know what they were. They didn't look like the leukemic cells necessarily, and so forth. And so then they stopped that, and then they said, well, let's just see if we can do this from normal cells. And so this is normal bone marrow cells that they would place into the culture. And Doris Morgan, because she was skilled in hematopoietic cell culture, you know, the immunologists, when they put lymphocytes into culture, they never looked at them under the microscope. They added tritiated thymidine as a precursor of DNA, and then they would harvest the cells and they'd analyze for the number of counts per minute of tritiated thymidine incorporation in a newly synthesized DNA. Well, that wasn't Doris Morgan's style. What she did was she looked at the cells under the microscope, and she babied those cultures, looked at them twice a day, maybe even 3 times a day. And she seeded those cells at a lower cell density than any immunologist would have ever done that. She put them at 100,000 cells per ml, 10 to the 5th per ml, whereas most immunological types would have put those cells in culture at 1 million cells per ml. That turns out to be very important. She put those cells into 20% fetal calf serum into Roswell Park Memorial Institute 1640 medium. And she cultured those cells either in flasks or in petri dishes so that she could then take them out of the incubator and then look at them under the microscope to see what was going on. And so I want to do some screen sharing in the results section. All right, so this is figure 1 of the data in this paper, and it's a growth curve of cells derived from normal human bone marrow. It's just one example, essentially. So on the y-axis, we have 10 of the total viable cells times 10 to the minus 5 per culture, per ml. And we have 2 different ones. We have control cells which is fresh medium, and then we also have cells seeded into lymphocyte conditioned medium. And you can see over the first week, to 6 to 7 days, both cells lose about 90% of the total cells seeded into these cultures. And then in the ones that got the lymphocyte conditioned medium, over the next week or so, there is an exponential growth. And then they split the cultures down again to 10 to the 5th. And then they go up again and so forth and so on. So they said that what they could do, this was just one example, but they could keep cells growing in this manner, in this lymphocyte conditioned medium, for 9 months, essentially. So let's take the screen share off. So that was the most important thing in this whole thing. In figure 2, which is the only other data in this paper, and I'm not going to show that because it didn't reproduce well from the journal article. They showed Cytospin, right Giemsa stain preparations of cells from these cultures at time 0, after 1 week, and after 3 weeks. And basically what happens is that almost all of the bone marrow precursor cells, which are mostly red cell precursors and PMN precursors, they die off over that first week, leaving behind lymphoid cells, both small lymphocytes as well as larger blastoid cells. But then by 3 weeks, all of the cells are blastoid, lymphoblasts, essentially. They were curious as to what kind of cells these were, and so them being hematologists, they stained them for all kinds of cytoplasmic and nuclear stains that might be able to tell them whether they were the lymphoid or myeloid. All the myeloid stains were negative. So, you know, this is back in the early days of immunology, and as we went through in the mouse, they had T cell markers, which was the theta antigen that subsequently became called, renamed Thi1. So they had antibodies to the surface molecule on T cells in the mouse, but we didn't have that in the human. Had to wait several more years in order to get that. But what they had discovered by then is that if they took sheep red blood cells, that the sheep red blood cells would stick to human T cells, and you would get rosettes. So they called them E-rosette. And they found that within a week or two, that 60 to 70% of the cells in these cultures that were lymphoid looking under the microscope and under the stains and so forth, those were Evozet positive. And then by 3 or 4 weeks, greater than 90% were Evozet positive. And by contrast, when they looked for B cell markers, surface immunoglobulin, they were negative. And that was important to them because they were worried that these were just EB virus infected cell lines, and that was not earth shaking. Everybody knew about that, essentially. So they're negative for surface immunoglobulin, and they were also negative for Epstein-Barr virus nuclear antigen, which was a tip-off that these these were lymphoblastoid cell lines as a consequence of EB virus infection. They were negative for that. And the final thing that really nailed down the coffin is that these cells, they wouldn't grow on their own in fresh medium. If you replace the conditioned medium with fresh medium, the cells would die in 24 hours. So they were absolutely dependent upon you feeding these cells this conditioned medium, whatever was in there was very special, essentially, special juice. So those are the data in this paper. In the discussion and in the summary of this paper, they make the point, they reinforce the point that this is reproducible. They've done it with 9 separate individuals, and they allude, they say, well, there must be a factor in this conditioned medium, but that's all they say. And they didn't reference in their paper, they only had 7 or 8 references. They didn't reference any of the immunological literature. Remember that we went through in 1965, it was reported in 2 back-to-back papers in Nature that there was something that lymphocyte stimulated conditioned medium would have a so-called blastogenic factor in it that would be mitogenic for other lymphocytes. That was 65. And then in 1970, Fritz Bach and his people talked about a conditioned medium reconstitution factor. And then Egal Gary in 1972 published those 2 back-to-back papers in the JAM talking about lymphocyte activating factor. Schimpel and Wecker talked about T cell replacing factor. So there were a lot of data out there that there was stuff, magic potions in this lymphocyte conditioned medium that made T cells grow, promoted them to grow. But they didn't reference this. And I don't think they ever read any of those papers, actually. They didn't know. They thought they discovered a whole new phenomenon. And what they really discovered, if you read this paper carefully, or not even have to even read it carefully, was that they showed very nicely in that figure that I showed that you can get cells to grow long-term, T cells to grow long-term in lymphocyte conditioned medium. That's what they showed. There's nothing about what the so-called factor might be that would that allow this growth. But often in the literature, this paper has been referenced as being the discovery of what became interleukin-2. And I've always had a little problem with that because they didn't discover interleukin-2 or T cell growth factor or anything like that. What they discovered, which was important, was that you could grow T cells in lymphocyte conditioned medium long term. The next question, the $64,000 question then, so what was it in this lymphocyte conditioned medium that was making them grow? And of course, that's where I spent the rest of my life in the laboratory. So that was it. That's published in September 1976. And subsequently, over the next few months, so Frank and Doris were working on this and so forth and so on, Gallo decided to just discontinue all work in his lab on these T cells and T cell cultures because he didn't want the lab to be distracted from what his primary purpose was, which was trying to find a retrovirus from acute myelocytic leukemia cells, AML cells. And so he basically called up Ron Herman, who was another investigator, but an immunologist at the NCI. And he just basically gave the whole project to him. He said, Ron, you take this. I don't want to, and he told both Doris Morgan and Frank that they were not to work on it anymore. Actually, Doris was just put to the bench to make more and more lymphocyte conditioned medium that then they shipped off to Ron Herman's lab. And so, as they say, the rest is history. Herman's lab had difficulty repeating the long-term growth of human T cells, and I wasn't surprised about that because we found it was much more difficult to get human T cells to grow long-term than it did to get mouse cells to grow long-term. And we were aware of all these experiments even before they published their very first paper, because Frank had called me up and asked me to do an interferon assay on the culture supernatants from these cultured T cells, just to see if that was that would be a criteria that they could make lymphokines. And we did those experiments and they were positive. And from there on after, I decided that this was a very important thing to work on. So we did, but we decided not to compete with Doris and Frank and Gallo. We switched everything to the mouse. And the reason being is that we'd already shown that we could generate cytotoxic T cells that would kill leukemia cells in vitro. And I wanted to see if we could grow them long term. That's going to be another paper that we'll get to eventually. So that's it. Thanks for tuning in. And don't forget, like it on the YouTube channel. We're getting more and more subscribers. I'm really pleased about that. Click on the bell so you get notified when we do a new journal club. And also go on my website and where you'll find all of the papers that I've decided are classics in immunology, as well as all my social media sites. So thanks for paying attention. It's been great.